Hello, everyone, and welcome to today's webinar, Highly Multiplexed Analysis of Mouse Neural Tissues. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Varenome Biosystems. To learn more, please visit their website at veranomebio.com. We encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our first speaker of the day, Chris Streck, the Director of Applications at Veranome Biosystems. Chris, welcome. You may now begin your presentation.
Thank you very much, and a warm thank you to the entire LabRoots team for arranging this. And I appreciate all the attendees for joining us today to hear about the Veranome solution for spatial transcriptomic analysis, as well as Dr. Hilbush's presentation of the mouse tissues. Again, my name is Chris Streck, and I'm going to give you a very brief overview of the Veranome Biosystems solution. As you have probably seen, with the advent of single-cell sequencing, there has been a drive farther in the direction to get higher resolution and a higher level of detail in analysis of individual tissues. And spatially resolved information has come to the forefront for that, for a lot of key investigations. In fact, in 2020, spatially resolved transcriptomics was identified as the method of the year by Nature methods, and we've seen a profound improvement in the overall technologies. Varenome Biosystems is a fairly new entrant to this realm. For those of you not familiar, Veranome emerged from a multi-year collaboration between Applied Materials and the Genome Institute of Singapore. This combined some of the world's leaders in engineering and semiconductor and display technology with a preeminent genomics research facility to develop new and powerful life sciences tools and the formation earlier this calendar year of Veranome Biosystems. Today, I'm going to talk to you about the overall Veranome imaging system, which includes an instrumentation that allows us to run very highly multiplexed spatial RNA analysis so that biologists can now look at subcellular resolution and leverage the expertise of the semiconductor industry's best experience and engineering for the highest quality and fastest results. As part of the Veranome solution, we've built a complete platform that includes upfront software tools to design your experiment, design your panels, track your experiment, and run and operate the actual instrumentation. Additionally, we've built an advanced imaging system that allows you to load your samples into a flow cell and obtain true single-cell resolution, even subcellular detection of messenger RNA molecules across a range of different tissues. We've designed this system to be very easy to use with very limited hands-on time and have true walk-away automation so that you can load your samples and then wait for the results to be generated. In terms of performance, we've spent a considerable amount of time to provide the highest quality data, focusing in on some of the newer aspects that are going to be key to resolving individual RNA molecules within individual cells. And we've built this system to be high throughput to match current lab workflows. Lastly, this is a benchtop system so that it can be a true democratization of spatial transcriptomics. Additionally, in addition to the software reagents, we also have a primary analysis suite that allows you to do both statistical and visual interpretation of our data. This is the portion of the presentation that Dr. Hilbush will go into detail, but it allows immediate actionable interpretation of the data coming out of the Veranome system. A quick overview of the Varenome Platform. This is a picture of the instrument, and we currently have multiple instruments in early access sites. The system is comprised of a flow cell loading chamber. All of the internal components are comprised of the optics for the actual detection and the fluidics control, as well as a built-in optical and optical table for vibration dampening. We've designed the system for easy access for sample loading. The individual reagents are loaded into the instrument in very simple rack formations, and the entire system is closed for, again, true walk-away automation. What we've designed this tool is to allow any researcher, whether they're working in traditional pathology or molecular biology, to conduct very highly multiplexed analysis for in situ tissue characterization. This includes pre-optimized assays and the panel content to allow you to have a single source for all of the reagents that have been developed and manufactured in a fully QC'd and controlled environment. This allows you to target hundreds of genes per run, and we've been able to demonstrate even larger panels. Additionally, this platform is able to do multi-omic capabilities with the ability to add protein targets, immunofluorescent protein targets, to your spatial RNA analysis, and then overlay and combine that transcriptomic and protein analysis into an individual experiment. Lastly, we've developed a number of different software analysis workflows using the state-of-the-art image analysis pipelines, use of traditional statistical analysis for data reduction or statistical clustering of the gene expression results, as well as a visualization tool to look at those combined results as an overlay onto the tissue image. In terms of the content for our platform, we've developed a system that's highly flexible. So we have the ability to have fully customized panels so that a researcher may access their own account and design their list of experiments and design their custom panels in a web-based portal. We are using our own proprietary design for the panel and the probe creation, and then you're also able to track your projects through the creation of your data and analysis. In addition to the fully customizable panel, we have pre-validated panels for both human and mouse in a number of different research areas, including immunology, oncology, developmental biology, and neurobiology. Additionally, we have the ability to mix and match modules. As the panels will grow in size, we will have basic core panels that will be comprised of the key usual suspect genes within a certain research area, and then be able to mix and match submodules so that you can provide off-the-shelf pre-validated custom panels using these subset modules. This will include specific immune cell markers to give better characterization of T cell subtypes or common tissue-specific stromal or oncology markers, as well as defining particular molecular pathways and functions. The combination of these three strategies for panel design give ultimate flexibility for researchers to design their experiments. Part of our solution includes a comprehensive software suite that allows researchers to plan their experiment, operate the instrumentation, collect their data, and analyze the results. So on the left-hand side, we see the instrument software that allows the researcher to set up their experiments and detail the sample-specific information associated with a particular run. We have a web application that allows researchers to design their targets or identify which panels they're using. And then we have an image analysis pipeline that automatically does all of the image processing, extracts the individual features, and quantifies the RNA molecules. Then individual biologists can take that data that was generated and visualize those results, conduct t-SNE, UMAP, or other clustering methods, and develop cell-based workflows that Dr. Hilbush will go through in subsequent slides. I wanted to give a quick overview just to highlight some of the ease of use capabilities of this system. So the entire workflow is set up within the instrument control software so that you can visualize your entire tissue, identify regions where you may want to do higher levels of interrogation, and then be able to set up the routine and track the project through the different stages of data collection. On the imaging processing side, we've leveraged a lot of the expertise for image analysis to develop novel methods and highly scalable methods for image processing, including segmentation. From the segmentation point, this is one of the most important components where we're using advanced algorithms to define the edges of the cells and quantify the number of spots in each individual cell. This allows us to get a large dynamic range for genes expressed both at high and low levels, and gives a true high level of biological complexity for high resolution of different cell phenotyping comparisons. We've also implemented some deep learning techniques for advanced cell segmentation that I'll show some brief examples of to tackle some of the greater challenges with defining cell boundaries. And we're doing this all with the expertise leveraged from our experienced software and engineering teams to make this very fast and very scalable. As we compare some of the segmentation algorithms and some of the improvements that the Varenome team has conducted, traditional segmentation oftentimes suffers from a lot of problems such as false negatives or false positives. And our deep learning algorithm is actually now been able to give higher accuracy in terms of detection of individual cells. This includes reduction of traditional false positives and artifacts, but also the ability to look at cells with different morphologies and cell sizes. What you see on the left-hand side is traditional segmentation, and on the right-hand side is the Varenome segmentation. And you can see very high resolution of cells with different sizes and different morphologies. Additionally, because there's going to be a wide range of tissue types coming into our platform, we've developed tools that allow researchers to customize or adjust the segmentation capabilities within their individual experiments. This allows researchers to set defaults, but then also adjust the segmentation boundaries to identify larger or smaller or more complex cell morphologies. This is going to be important to identify the maximum number of spots, RNA spots, associated with any particular cell region. In this particular example, the light on the left-hand side, the light blue was the default boundary setting from the algorithm. And through the hands-on and user-adjustable boundaries, researchers can then tweak a number of different components to adjust those boundaries to result in larger or more accurate cell areas. So I've tried to give a basic overview of the system, but I'd like to highlight a couple of the different application areas where we're focused. So one of the key applications that was demonstrated at this year's AGBT conference by one of our research collaborators at the Genome Institute of Singapore was to look at organoids at subcellular resolution, looking at anywhere from 5,000 to 50,000 cells per tissue and as high as 547 genes. This was a very exciting talk that demonstrated the true biological utility of the Varenome platform. Additionally, through our collaborators, we've got a number of different experiments, building off of that initial, some of the initial technical development, not just with the human brain organoid, but also with a considerable amount of mouse neural models as well as human brain tissues. What I'm seeing here what I'm showing here in the two middle panels represent some of the mouse neurotissues that Dr. Hilbush will go through, but also on the right-hand side, the human brain tissue that represents both the RNA as well as protein markers simultaneously captured in the experiment. Additionally, through our collaborations, we're working with a number of different IO researchers to profile oncology samples and get the true heterogeneity associated with the tumor microenvironment. Lastly, I want to finish with just a couple notes about Veranome as a company. So at AGBT this year, so in March of 2021, we announced the formation of the company and specifically talked about our early access placements that have been going on for quite some time. Up until this point, we had been quietly working on a number of different pilot studies through our service program and are expanding that capabilities through a variety of different manners. Additionally, last month in June of 2021, we announced a co-development partnership with Portal Biosciences. This is to implement a second-generation assay to allow us to interrogate challenging samples such as FFPE compatibility, as well as get higher resolution of your RNA targets, including splice junctions and all the way down to single nucleotide resolution. Lastly, as we've described in previous press releases, we are in the process of our expanded beta program and working towards commercialization. This includes complete infrastructure to provide fully developed QC'd and robustly manufactured reagents, software, and instrumentation. So hopefully today I've been able to give you a brief overview of the system. We'll end the presentation with answering questions, and we'll provide some additional information on how to get in touch with the Veranome team and get additional detail. With that, I'd like to turn it over to Brian.
Thank you, Chris, for that presentation. I'd like to introduce our second speaker of the day, Dr. Brian Hilbush, Director of Digital Solutions at Varenome Biosystems. Brian, welcome. You may now begin your presentation.
Okay, great. Hello, this is Brian Hilbush from Veranome Biosystems. In this part of the webinar, I'm going to talk about highly multiplexed analysis of mouse neural tissues. We are developing a set of robust applications for neuroscience research, and I'm going to show you examples of our data that highlight the strengths and features of our system. And to start, just a little background on neuroscience. Neuroscience is a highly integrative discipline, and for the past 100 years has really involved both neuroanatomy, neurophysiology, and genetics to get to an understanding of nerve function. And in the last couple of decades, it's really been computational methods and genomics that have really propelled neuroscience forward. And so at Veranome, we've produced a highly multiplexed spatial omics system that I think is going to really accelerate research. And I'm going to show you that through a series of examples now. And one of the things that we have done with the system, as Chris described, is to build multiplex panels that really can address a number of experimental paradigms and evaluations in neuroscience. These key applications include, looking at cellular responses to drugs, experimental perturbations, CRISPR screens, and other aspects of pathobiology. It includes being able to look at spatial dynamics and do careful pattern detection across, brain subregions. And then finally, we're looking at a lot of applications that involve doing cellular phenotyping, transcriptional phenotyping, and just evaluating cell states as well as neuroanatomical mapping of cell types. So the Varenome system generates beautiful data for this. We have analytics that are tightly integrated to the data types that are being produced and required for these types of analyses. And they lead really straight to biological insights as sort of defined on the right side of this slide. The panels that we have designed, the first one that we did was to really cover synaptic function and have just general markers for neuronal and glial cells in the mouse brain. So these were heavily weighted towards neurotransmitter receptors, neuropeptide receptors, a variety of ion channels, as well as a whole array of transporters in the cellular carrier family. So those have been very useful in being able to do some initial explorations from R&D sort of standpoint. And we've now built a new panel that we call Mouse Neuro V2, which is a lot more balanced and has a much wider variety of cell type specific markers as well as universal markers of a range of cell types as indicated here on the left. And then those cover a variety of biological processes and ability to kind of sort of surveil pathological states through this panel composition. So those, I'm going to show you mainly examples in this webinar around the Neuro V2 data panel. One of the things that really drives the system is our integrated informatics that we have built. We have tools that are designed to really walk an investigator through 3 different workflows. One that's simply taking data, sort of a gene-centric view, driving a menu of genes that are on the panel, doing quality control checks, and being able to explore the data quite interactively in the visualization screen. We then have another workflow that allows initial explorations of patterns and to do some number crunching around correlation and sort of binning and looking at spatial networks. And then finally, we've got a real unique workflow designed around single cell based clustering that we can do really fine expression signature analysis, produce a variety of plots to look at the data such as t-SNE and UMAP, as well as being able to sort of drill down and do some fine filtering and selection as needed. So one of the starting points, I think, for showing you the power of the system is to give you some examples from the mouse cerebellum in which we have done a preliminary survey. This really gives you a feel, I think, for how sensitive the transcription detection is, the ability of us to really pull out highly specific cells with the first panel that we built, and then to really give you a feel for just sort of the multiplex visualization that these panels provide combined with the software, and you'll see the patterns come out. So the bottom left there is just an image from the mouse brain from the Allen Brain Atlas, showing you kind of the region of the cerebellum that we section, and we're going to look at data through sagittal sections. For those of you that aren't familiar with the cerebellum, it's a very simple structure, very beautiful. It has a simple 3-layer structure. The top layer, pictured on the left and the right, are the molecular layer, where the Purkinje cells extend their processes out into and are contacted by a variety of cell types, including the granule cell climbing fibers, as well as interacting with basket and stellate cells in that layer. Purkinje cell layer has both Purkinje cells as well as astrocytes like the Bergman glia, and then also the granule cell layers densely packed with granule cell neurons and other support cells. So the picture on the right shows you sort of the first glimpse of just how sensitive our technology is. If you can look at the sort of the turquoise overlay, you'll see the inositol triphosphate receptor highlighting high RNA expression way out in dendrites and Purkinje neurons that are projecting out into the molecular layer. This is really one of the most striking things we've seen in this analysis of the cerebellum. So we have a number of examples to kind of give you a little bit more on, sort of how we're able to get this resolution. And this is clearly subcellular because you're looking at processes that are extending out that are probably submicron in size. Just a bigger blow-up picture of this for you. On the left-hand side, you can see Purkinje neurons dotted with that RNA expression. And you'll see a few empty spots along the way there. On the lower left, you can see that they're actually occupied by the Bergman glia, which are expressing high levels of this transporter. So that's kind of a beautiful example of kind of the exquisite cell type specificity of these markers, as well as the ability of the system to provide you very high resolution imaging across these cells. And on the right is just another view of these two. Going into other cell types and other molecular classes, looking at glutamate receptors such as the metabotropic glutamate receptor, you can see it's very highly expressed in the granule cell layer, much lower in the molecular layer, in contrast to the AMPA receptor, GRIA4, which is primarily expressed out in the molecular layer. So just a couple of pretty pictures to show you sort of what we're seeing as we look through just a couple of markers in the cerebellum. A couple of other examples are a potassium channel, a delayed rectifier that's expressed, looks like mainly in basket cells and stellate cells. Gives you a really nice view of sort of how they're distributed out in the molecular layer. And then finally, in white matter regions, you can see clearly myelin basic protein, as well as aquaporin for astrocytes being distributed out in the the fringes there. One of the really nice things that what you can do is you can move from visualization right into expression correlation analysis. And here I'm just showing you some data from looking at a subset of the receptors that we've got on this panel, and you can see right away that there is high correlation of spatial expression of a number of GABA receptors, including GABA B 1 and 2, along with the IP3 receptor, and also the serotonin, one of the serotonin receptors. So it's really a very quick and easy way of looking at expression profiling, sort of at kind of a co-expression sort of level. Of course, the system can do a lot more than that. One of the powerful things that I think the system provides is the ability to generate molecular signatures and beautiful expression maps from really what we've seen now are a variety of structures that are quite diverse across the brain, including hippocampus, the cortex, which is quite layered, as well as midbrain, hindbrain, and other regions in the mouse CNS that are much less structured, but have underlying structure of the nuclei, and we'll see some examples in a minute here. But this slide is just showing you examples of the signatures that are generated from these structures. The top part is the signature off of the V1 panel for this region on the left, and you can see the array of markers. And on the bottom is the molecular signatures from the V2 panel, and there you can see that right away the cluster analysis and the panel is able to discriminate the number of neuronal cell types, glutamate neurons versus GABAergic interneurons, a couple of oligodendrocyte classes, astrocytes, as well as microglia, endothelial, and another couple of subclasses of oligodendrocyte and neurons. So right away with the panels, we're able to get really clear distinctions on cell type just by looking at the detection rate, which is indicated by the strength of those dots. So the smaller dots indicate lesser expression and a lower detection rate on a per cluster basis versus really large dots, which are cells that have very high detection rates. There's also, you're looking at the Z-scores as well on the bar on the right. You've got Z-scores just distributed from negative to positive, and you can see cluster representation and the variance that helps define those clusters using that Z-score information. All that information can be downloaded into Excel spreadsheets if need be, or put into other programs. So yeah, the export of Veranome data is quite simple. It looks just like single cell data, so you can export it into programs like Seurat. This is a little bit of data from our mouse striatum example. Here I'm just showing you sort of the basic steps that you can take in Seurat to go from normalized and scaled data to principal components analysis, and then you can generate heat maps as shown on the top there for the different principal components in these cell by gene matrices. And then also you can generate t-SNE and UMAP plots in that analysis package as well, in addition to getting sort of biomarker statistics off of the most highly represented genes in your particular sample. So with the NeuroV2 panel, I think one of the most beautiful things is that we can really begin to see a much broader clustering of these markers across your analysis. Here I'm just showing you an array of marker types and co-expression patterns ranging from the neuronal types at the top there, glutamatergic, GABAergic, and just general neuronal universal markers, all the way down through much more subtle classes like the neural stem cells in the middle, OPCs, oligodendrocytes, and so forth. So this panel really is equipped with a range of tools to interrogate neuronal systems, and the software itself can then provide alternative views, and spatial maps, and ways to do sort of correlation analysis. One of the things that we have done is just to simply look at the data and see what it tells us. And one of the things that is striking to me is that given these panels and the clustering tools, one can actually begin to see substructures in the brain that are sort of only evident by sort of careful neuroanatomical mapping. So with the cell typing ability and the clustering, you can see subdivisions in the thalamus quite clearly, as indicated on the left-hand panel there. So hippocampus is obvious. The thalamus not quite as obvious, but you can see clearly, right next to the lateral ventricle, sort of fiber tracks going through the thalamus, right next to the reticular nucleus, and then a number of areas in the thalamus that are projection neurons that are glutamatergic, and that's sort of the defining parts of the thalamus there. On the right-hand side, I just picked one example of one gene that tends to highlight mostly the glutamatergic neurons and gives you a nice definition of the thalamic nuclei, and that's VIPR1, the receptor for the vasoactive intestinal peptide. And you can see down on the lower right-hand corner just sort of the strength of expression of that particular RNA in the thalamus versus other brain regions. If we look at it a little bit more closely, what's really interesting is that the system is able to produce some insights around sort of subregions that have not really been molecularly defined before. So on the left-hand panel, you're seeing clusters of GABAergic neurons highlighted in white there. You see some streaming out there in the cortex, a little bit in the subiculum, and then down on the lower part of that left-hand panel, you'll see the reticular nucleus on the left and the right-hand side. The Viper is just there for reference. In the middle, I've found genes such as FGF1 that highlight the reticular nucleus pretty well as a glutamate receptor, GRIC3, that's known to be expressed quite highly there. On the right, we're seeing some kind of novel findings with markers such as CD6, and then also a couple of other genes, ITGA7, CDK6, as well as some kinases that we've identified in doing the analysis. So there's a small breakdown of this particular region into very small subregions that do or take different inputs and provide signals that are used to regulate the thalamus through GABAergic output, negative output. And so this is probably indicating there's one of these regions that's being highlighted by this molecular signature. So that's just an interesting observation to show you sort of the power of the system. Another thing that we have spent time on is to just demonstrate that we can get high resolution cell type specificity of markers that are of importance in doing cell typing analyses. In this example, we're looking at the hypothalamus, and in particular, zoomed in on a region of the hypothalamus known as the lateral hypothalamic area. It has both glutamatergic and GABAergic cell types that are well known. And actually, there's quite a number of subtypes of those as well. So if you start in the upper left-hand corner, that's kind of the higher, lower level view, sorry, of the region that was imaged. If we cluster that and get clusters on the lower left-hand side, lower left-hand panel, and then move over to the kind of the panel in the middle there that's highlighting the two different neuronal types, we can actually do some quantitation of how well the markers are performing just by counting, in this case, a few hundred cells and investigating sensitivity and specificity. On the right-hand side, I've counted the cells or two markers for glutamate neurons as well as for the GABA neurons, and you can see the sensitivity for the synaptic vesicle transporters, is quite good in the mid-80s. And then for the GABAergic neurons, the synthesizing enzyme GAD2 is super high, with almost 100% sensitivity or recall. So that's really a really interesting finding. I think one of the things that gives us a lot of confidence around being able to use these as markers kind of throughout the brain. We've also done these sorts of investigations in the human cerebral cortex, and this is just again looking at the GABAergic and glutamatergic neurons, highlighting the fact that you can discriminate these cell types, quite easily in the brain. And I'm showing you an overlay that the system has to just really display both protein markers as well as the RNA markers. The protein markers we're showing here are nu-in for neurons in green, and then the astrocytes you can see clearly in blue. So it really looks like we're doing quite well in terms of how clearly localized the expression patterns are. It's really one of the most interesting things about the system is that we can really turn this into a multi-omic tool. Again, just doing a small bit of analysis on the human cerebral cortex. You can see on these slides here that neurons, the neuronal clusters are highlighted in blue, the astrocytes in red, and you can see on the left sort of the immunofluorescence with GFAP, which is a strong astrocyte marker. And you can see the red groupings are RNA-based markers that are overlying most of the astrocytes. Blue is for the neurons, so a little bit higher resolution view of that on the right. So those are sort of some of the features that we've got with analyzing neuronal types particularly in the mouse brain. And this is really sort of giving you sort of a feel for just what the system can do. It really gives you a validated high-resolution spatial platform for really a variety of neuroscience applications shown down below there. The data that's generated really is that single-cell resolution, and you can accurately resolve, brain region patterning and dive into, the complexity of what's happening in the nervous system and really evaluate the heterogeneity. The panel designs that we're working on really offer a powerful solution for doing, cellular phenotyping across a range of models in neurodevelopment and also the multi-omic analysis capabilities. So the experiments that one's most focused on in neuroscience today, on the left there, we can generate the data. We have the analysis tools to quickly work with, our platform's data and then generate the insights kind of listed there on the right. So that's a quick run through of the mouse brain data. And I wanted to thank the entire Varenome Biosystems team, which has done a tremendous amount of work in just producing the instrument, producing the data, and also all the beautiful analysis tools. So with that, I'm going to leave you with just a request to check out our social media sites. Follow us on Twitter and LinkedIn. And now I'm going to take some questions, from the audience at the end of the webinar. Thank you.
Thank you, Chris and Brian, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, Please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, your data with glutamate and GABA markers for discriminating neuronal types showed impressive sensitivity and specificity in the cortex and hypothalamus. Do you have data on other markers that perform as well?
Yeah, hi, thanks, Alexis. Yes, we do. We've generated, analyzed, and validated probably over 50 cell type specific markers. The validation has been done using examination of cellular and spatial expression patterns and by analyzing their performance in clustering sort of experiments. So these include additional neuronal cell type markers in cortex, striatum, thalamus, and other regions. And we also have a series of excellent markers for oligodendrocytes, subtypes of oligos, microglia, both activated and just homeostatic, as well as endothelial cells and astrocytes. We also have these for both human and mouse panels.
Great. Thank you so much, Brian. So our next question looks like it is a two-part question. Is the system doing the imaging, or do you need an outside imager?
Hi, sure. So the system is entirely self-contained in the instrument. So the imager and the microfluidics are all contained in a single benchtop system. So you don't need a separate instrument to do the chemistry.
Great, thank you, Chris. We're going to go back to Brian for this next question. Is Veranome cell workflow, what tools do you have to track cell type heterogeneity within brain regions and across regions?
Yeah, that's a really great question on heterogeneity. I think, since we have the single cell resolution, we can examine the entire cell by gene matrix and determine subsets of cells of interest and look at their expression patterns. From the matrix, you can look at expression signatures or normalized expression values. And these can be compared for cells that are adjacent or within the same brain region or even across different regions. And since we have the spatial information, we can actually select cells or regions from the image and examine heterogeneity that way at a very high level of resolution. And do so around, say, neurodegenerative plaques or areas of inflammation. So it's a lot of great tools for that.
Great, thank you. Our next question. In the thalamic reticular nucleus example, you showed evidence of a sub-region specific signature. Have you been able to see other potentially novel signatures across other brain regions?
Yeah, again, this is being used pretty well as a discovery tool, and we've been able to detect novel signatures, across other brain regions. And we're looking at really interesting results in subregions of the cortex, the hindbrain, thalamus, where we're seeing really novel patterns and expression signatures. And the data, suggests potentially new cellular subclasses or at least novel functional states that are unique. And again, since we have both quantitative differences in expression and spatial patterns, we don't necessarily have to rely solely on the numbers like you would do in a single cell sequencing type experiment.
Thank you so much. And I do want to remind our audience, we've gotten so many great questions in today. Any questions we are unable to get to today, we'll answer via email with the email address that you used to register for today's event. So our next question, can your cell segmentation parameters be adjusted and optimized by the user?
Yeah, I can take that one. Yes, indeed. I mean, the cell segmentation component, prior to looking at the data, users are going to have access to an application where they can select a type of algorithm, the training model, and also parameters such as dilation, memory dilation, to optimize performance. So our R&D team is building and validating segmentation algorithms. And currently we're outperforming segmentation algorithms such as CellPose, and we're going to continue to sort of improve on our deep learning approach with nuclear and cell boundary markers. So we've got a lot of activity in that area, and we think we're probably at the forefront.
Great, thank you so much. Now, I believe this may have been mentioned in the presentation, but just to confirm, Can this be used on human brain tissue?
Oh,
yes,
yeah.
Yes, we're currently doing a lot of, oh, sorry, go ahead, Brian.
Yeah, absolutely. I mean, we've got a lot of applications coming in on the human brain, and we're actively generating data on that right now.
Yeah, and one of the panels, one of the experiments that I showed in the early presentation was, we didn't do a deeper dive, but we'll be doing a follow-up webinar on the human tissues. But one of the panels that I demonstrated was a highly multiplexed RNA target plus co-detection of a number of different proteins, NEUN, and a couple of other key markers. So we're quite a bit of our early collaborations are in the human neuro realm.
Thank you, gentlemen, both for your answers. Now, how does the algorithm produce higher accuracy in segmentation?
Chris, do you want to handle that? Or do you want me to address that?
Sure. So I'll let you handle the technical component. Another part of our webinar series is going to be walking through our image analysis process, including the segmentation results. And what we've been doing is benchmarking our algorithm to some of the open source or published segmentation algorithms out there. So with that, I'll let Brian tackle the next step.
Yeah, I mean, I think most of the superior performance we're getting comes from our deep learning framework. So we use a convolutional neural net and some other tricks to sort of really eke the highest performance out. And we have really good annotation data sets with which we can do the training and also the benchmarking. So yeah, we've got a lot of tools for that.
Great, thank you, gentlemen. We've got time for a couple more questions. So our next question is, do you clear the tissue?
Sure, that's a great question. A lot of the early work that we've been doing with our collaborators is to bring in a host of different tissue types, different qualities, that sort of thing. As you probably know, different tissues will have varying levels of autofluorescence or varying levels of overall background noise. And so we've developed a number of different sample prep protocols and techniques. So some methods, some particular tissues, the clearing is not required. Other avenues, other tissue sources, we have been working on implementation of such methods. It really comes down to characterizing and providing our customers with a decision tree to help them understand which path they want to follow.
Thank you, Chris. And I believe this question is going to go to Brian. So can you describe the time it takes to go from image generation to data analysis?
Yeah, sure. There are two stages really to get analysis ready data. The first is to take a reasonably sized region of interest, say hippocampus and thalamus, and then take that image and perform calibration, registration, and decoding, plus the cell segmentation. So all that pre-processing steps, all those steps take probably somewhere in the range of 1 to 3 hours. The second stage is just simply loading the processed image file into the Veriscape browser. The file is usually read in and ready in a few minutes. Analysis can be undertaken immediately. There's really very little data preparation and downtime, and the desktop application will do and process algorithms to produce t-SNE plots and clustering and image rendering almost in near real time for most of that. So that's the sort of overall time frame, going from sort of imaging on the system to analysis probably within 3 to 4 hours.
Great, thank you so much. And Chris, I believe you may have had a comment for the audience real quick.
Sure. So we've got a number of questions asking about the overall throughput image area, as well as the sensitivity of detection. So rather than answering each one of them, I might just tackle them all in one answer, because there's a lot of repeat questions. So in terms of the sensitivity, we're on par with single molecule detection associated with standard SM fish. So the the technology is uniquely sensitive for detection of individual molecules, but as we know through the development, it also requires, to get high quality data, it also requires being able to discern individual RNAs next to each other. And so that's really where the technical team has shown and developed a very high quality product. In terms of the plexity of the panels, we're working typically in the hundreds currently. We've demonstrated higher, but we're typically in that sweet spot for most experiments these days. And then on the image area, as you saw from one of my slides, we've got the whole. We're starting with pieces of tissue. I think you could have visualized pretty much the entire mouse, the entire mouse brain, and then the overall area that you're imaging is going to be dependent on how much you want to see, so that's tunable to the researcher, and the number of plexi that you're looking at. And so it's more of a matrix for how long an experiment will take based on those parameters. So it's kind of there's not a straightforward, this is the one size fits all type of run. It's tunable to your particular experiments. And I'd recommend anyone who's interested in those, in additional details, we're happy to set up, answer those questions in individual calls or follow-up sessions. And Alexis, back to you for other questions.
Great. Thank you so much, Chris. Looks like we have time for maybe one, maybe two more questions for here today. So, and now Brian and Chris, I believe this is going to be a question for both of you, and it is a two-part question. How long does the assay take? And the second portion is, how long does the analysis take?
Sure. So the overall experiment requires a fairly short amount of time, hands-on time, to set up the individual assay. So once your tissues have been sectioned onto our slide, the overall bench time is really a very quick process. So there's some hybridization steps. Once the flow cell is set onto the instrument and the instrument runs, all of that portion is automated. So that our instrument runs can be anywhere from just a few hours to over 8 hours. So it's a pretty rapid turnaround in terms of the total time. Additionally, multiple samples can be prepped at the same time, so that if you wanted to set up multiple runs in serial, your bench time can actually be, your bench time for sample prep is actually a parallel process to drastically reduce the typical workflow time. In terms of the analysis, I'll let Brian handle that question.
Yeah, the analysis time is really pretty rapid. I mean, I think within 15 to 20 minutes, you can probably get, cluster visualization, t-SNE plots, looking at cell by gene matrix numbers, and doing a lot of visual inspection of, molecular maps and that sort of thing, and looking at correlation data, very, very quickly. Offline, you could probably then download the data into other, statistical analysis packages and do whatever you would like. So, you're actually getting to data very, very quickly on the system with the tools that we've already built in.
Great. Thank you so much, Brian and Chris, for answering all of these amazing questions. Do you have any final comments for our audience? Chris, I'll go ahead and start with you.
Yes, thank you very much for hosting this and being our Sherpa through the question and answer series. And I'd like to thank the audience for joining us today. Again, we're happy to answer any additional questions. If you'd like additional information, please visit us at varenomebio.com or send us an email at info@varenomebio.com, where we'll be happy to follow up. Additionally, we've got a series of webinars that we'll be posting on our website as well. So please join us again to see additional biological insights, as well as additional details on the chemistry, the assay, and the overall system. Yeah, so thank you all. And then, oh, sorry, go ahead, Brian.
No, I just want to say thanks to the audience, and we'd love to be in contact with anyone that has interest in neuroscience applications and that what the platform can do for you.
Great. Thank you again, Chris and Brian, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Veranome Biosystems, for underwriting today's educational webcast. Before we go, I would like to thank the audience for joining us today and for their interesting questions that we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, bye-bye, everyone.